Hello and welcome to Giants of Gene Therapy. I'm Hans-Peter Kiem, President of the American Society of Gene and Cell Therapy. My guest today is another true giant in our field and pioneer in the fight against cancer, Dr. Carl June. Carl is the Richard Vague Professor in Immunotherapy in the Department of Pathology and Laboratory Medicine at the University of Pennsylvania. He's also the Director of the Center for Cellular Immunotherapy and the Director of the Parker Institute for Cancer Immunotherapy at UPenn. Carl has pioneered the development of CAR T cells and the use of genetically modified T cells. He was truly instrumental in bringing CAR T cells to the clinic and also received the first FDA approval for CAR T cell therapy in 2017. In 2018, Carl was featured in Time 100 as one of the most influential people in the world for the invention of CAR T cell therapy for cancer. Dr. June is a prolific researcher who has published more than 500 manuscripts. He received numerous awards and honors, including his election into the National Academies of Medicine and Science and the American Academy of Arts and Sciences. He's been the founder or co-founder of quite a number of companies involved in T-cell therapies, and Dr. June has been an ASGCT member and annual meeting keynote speaker. Thank you so much for making time today, Carl. My first question will be, tell us a little bit about where you grew up and what got you into science very early on.
Thanks, and also thanks, Hans-Peter, for the introduction. And I grew up in the San Francisco Bay Area, and my dad was a chemical engineer. I was admitted to Stanford, in 1971 at completion of high school, but then that was right in the midst of the Vietnam War. And I ended up getting a low draft number. And so I went into the Navy and never actually went to Stanford. So I moved from the West Coast to East Coast then.
So in your family, was there science? Did you really grow up, talking about science?
No, well, my dad was an engineer and mathematician. So but there was no science, and there'd never been a physician in my family.
Okay.
And I started off thinking I would be a chemist, like my father, but then got really interested in, biology, and that led to medical school.
Obviously, you were accepted to Stanford, but then you went to the Naval Academy, correct?
Yes, yeah.
About that transition, how that all came about.
Yeah, it was one, I have to say it was completely unexpected. No one in my family had ever gone into the military like that. And so it was, 4 years of undergraduate where I did, chemistry and biology. And then the war got over, the Vietnam War, and I had a 7-year obligation, and they opened up opportunities to go to medical school. And so, I was given a scholarship and went to medical school in Houston at Baylor College of Medicine, all funded by the Navy.
And then after medical school?
Yeah, well, I had then, at that point, they had put me through 4 years of undergraduate on scholarship and then medical school. I owed 12 years back to the Navy. And, through medical school, I had gotten interested in immunology. My mother had lupus when I was growing up, and autoimmune disease runs a lot in my family. So I began working in medical school in an immunology lab studying immune complexes. And then I was able to go to Switzerland for a year and work in the World Health Organization. And I worked there on the immunology of malaria. And that's my first publications. So I got very interested in immunology, in medical school, but the Navy wouldn't let me go and get a PhD. So I was not allowed. I tried, and they didn't. All they wanted was, the medical training. And so I was able to get a whole year of work as a graduate student in Geneva, Switzerland at the World Health Organization. And then I was very fortunate to go to the Fred Hutchinson Cancer Center for bone marrow transplantation therapy, your institution, where I then, continued in immunology.
Tell us a little bit about that time at the Fred Hutch, of course, when it started out, and before my time.
I was very interested in immunology and, in infections. I had been, working on malaria, and then HIV, came out right then as I got done with my internal medicine training. And so the Fred Hutch was really the birthplace of allogeneic bone marrow transplantation, as you know. It's really the first cell therapy. And that's really what, and there I studied T cell activation. I was in with Paul Martin and John Hansen, and got very interested in the activation of T cells because I saw these horrible cases of graft versus host disease. And we knew from the mouse studies that many people had done that T cells are what are doing this, orchestrating immune destruction. And so I became interested in how we could activate and control T cells. And so that led to studies on the biochemical signal transduction of the T cell receptor and co-stimulatory molecules like CD28.
This was done right during the kind of mid 80s, if I remember correctly, at that time. You also worked with Craig Thompson at that time, right?
Yeah, and that's an interesting story. Craig was also in the Navy, and so he had gone to Dartmouth Medical School on a Navy scholarship while I had gone to Baylor. Then the Navy decided, and this is another interesting story, the reason the Navy decided to train us for bone marrow transplant, they trained 4 physicians to do allo bone marrow transplants in 1983, way before it became, generally into medicine as it is now. And the reason they did that was the Navy had nuclear submarines, which could lead to limited nuclear casualties, as you know, from radiation issues. And actually the Navy in the midst of this time it was a Cold War. And with Russia, the Navy needed ships that could go up through Japan, and Japan actually required that we have a program to treat nuclear casualties. And so that's why Craig and I ended up at the Fred Hutch. And that's where I met him. We worked very closely for 20 years together, and it was all because of the Cold War.
And so, but then the Navy, right, you've mentioned before, and you had to focus on infectious diseases. And then, of course, there was HIV. And of course, that's also how I got to know about your work with HIV. Maybe tell us a little bit about, your thoughts at that time, in terms of, how to harness the immune system to fight HIV.
Yeah, and that's another interesting story and another unplanned fork in my career. So the Navy, there's, the U.S. government, as you know, funds cancer research through a congressionally appropriated funds directly to the National Cancer Institute. And the Navy, in fact, all the armed services, Army and Navy and Air Force, are only allowed to do research in infectious disease and in combat casualty care, or, like trauma. Mm-hmm. So I was trained, as you know, as an oncologist at the Fred Hutchinson Cancer Center. I was board certified. I was a bone marrow transplanter, and I couldn't do research in that field as long as I was in the Navy. So I worked, and began working on HIV. And my first paper ever published in Science was on HIV and what it did in integration, latent HIV, and so on. So. And that turned out to be really useful later with CAR T cells, because I learned a lot about the virology of HIV and how to use it then, as you know, as a lentiviral vector. So it was all due to that I was not starting out my research in cancer therapy, which is where I had been initially interested. But
you started also working on CAR T cells in HIV. That was one of your first studies, right?
Yeah, and that happened because one of my I had Bruce Levine came as a postdoc in my lab, and we were studying HIV immunopathogenesis, had a paper about this latent effects of what HIV does to T cells that survive and end up having latently integrated HIV. And in doing that, we were studying and trying to grow T cells from patients with AIDS. This is before potent antiretrovirals were around. So the patients all had high viral loads and very low T cell counts. And no one had ever really gotten T cells to grow well from those patients. And they had they died from immunodeficiency of, T cell lymphopenia. And so we wanted to quantitatively improve and qualitatively improve their T cells. And Bruce Levine did an experiment where, we had worked on CD28 as activating T cells, put them on beads, and it works very well to grow cells. And so we started using that on HIV. And to grow CD4 cells from these AIDS patients. And we had this interesting thing where we had to make viral stocks of HIV, and we wanted to get natural, HIV from directly from patients rather than having lab adapted strains. So I told Bruce, go take, we had HIV blood donors, just take one of those patients, and then grow up the T cells, and then the virus will come out, and then we can use that as a stock. Well, he did that, and with the CD28 coated beads, the cells grew great. And there was no virus came out, in spite of a lot of replicating, CD4 cells. And we had control experiments where if we activated with lectins like PHA, plenty of virus came out, lots of cytopathic effects. And that ended up being a paper in Nature because we found out that CD28 downregulated CCR5, co-stimulatory receptor, a co-receptor for HIV. And CTLA-4 has the opposite effect. It turns up the receptor. And you can really make HIV much more pathogenic that way. So that's how we found that out. And when we had that paper published, Cell Genesis, which was a company in South Biotech in South San Francisco then, saw that we could grow T cells efficiently. And they made a CAR called CD4 Zeta for HIV. And so we partnered with them to do the first trial in patients, started in the late 1990s with CAR T cells in HIV infected patients.
Yeah, I do remember that very well, actually. Yeah. So this was also around the time when other people were working on or sort of started, I think, thinking about CAR T cells. I think one of the other really people was Zelig Ashar, right, who was really, working on that concept, even though not necessarily for cancer at that time.
Yeah, Zelig, so it's a very interesting story, really, because it shows how basic science can lead to unanticipated clinical benefits in medicine. And so Zelig was testing haptens and wanted to see if he could get MHC independent activation of T cells in mouse T cells. And so he made, grafted onto the T cell receptor. So not exactly the same design as we use in a CAR molecule now, but he could then show MHC independent activation of T cells. That was in 1989. And actually what people was missed was that a lab in Japan had done that in 1987, a Professor Kawana, and published, and there they showed in a little cited paper in BBRC that you could also get MHC independent signal transduction through the T cell receptor. By grafting on antibody fragments to the TCR. So then Zelig in 1993 published a paper in PNAS where he then made an independent molecule. Instead of grafting onto the TCR, he put antibody binding domains onto the zeta chain of the T cell receptor, and he called those T-bodies. And that led then, to many people working on these as an MHC independent way to redirect T cell specificity.
So who else was working on that concept at that time? This was like then around the 90s, early 90s, I think, right?
It was about a handful. Well, a couple of things constrained it. There were about 5 labs working on that. Where I started at the Fred Hutch, Phil Greenberg laid many of the principles for, adoptive cell therapy and studies in mice, and he mostly was interested in T cell receptor redirected cells. But Ollie Press, who was a fellow with me while I was there with Craig Thompson, became very interested in retargeting T cells with rituximab. And so he made CD20 CARs. And then he worked with Mike Jensen and Lawrence Cooper, who was in Phil Greenberg's lab. And so they had a nucleus starting there at the Fred Hutch there. And then Michelle Sadelain had a very active and long-term interest in that at the Memorial Sloan Cancer Center. And then Zelig Eshar went and did a sabbatical at the National Cancer Institute, where he worked with Patrick Hugh and Steve Rosenberg to make CAR T cells targeting folate receptor alpha in ovarian cancer.
So this was then around the time when I think people realized, right, to use gene therapy to really retarget the immune system. Because
Yeah.
Steve Rosenberg had done, tumor infiltrating lymphocytes in many studies, right? I know we've done here many studies initially with unmodified T cells.
So that's exactly right, Hans-Peter. What happened was the confluence of, genetic engineering, gamma retroviruses and lentiviruses to allow gene insertion efficiently into T cells. The way to grow T cells efficiently, which, we found from my lab, the beads activating through CD28. And then, the use of co-stimulatory domains, because Zelig-Eshard only used the zeta chain of the T cell receptor, and there was no co-stimulation that, so to get long-term persistence of cells, it was necessary to use and add in molecules like CD28 or 4-1BB in, so-called second generation CAR T cell designs. But what a lot of people also don't realize was that, but you certainly well know, is, the accidents in gene therapy, the well-publicized death of Jesse Gelsinger in 1999, that made all the biotech stop, not believe that it was possible to do gene therapy.
Mm-hmm.
So just the labs kind of that we've been discussing continued academically, but biotech, basically at that point, gave up on that approach. So it was carried on in an academic environment by the people we're talking about. And that's the only way it survived.
That's right. I remember that. Now, it was very hard to actually get money, right, for these studies and to fund, they looked very promising, but there was really no funding there. So how did you survive those times?
I mean, it was really, I think this is, the story is Michelle Sadelain and I really were the first to do co-stimulatory, second generation CAR T cells. We did at Memorial with a CD19 CAR and CD28, and that became basically what Yescarta is today from Kite Gilead. And I did our studies with, 4-1BB, CD19 CARs. And, but that all happened because of unappreciated thing from Barbara and Edward Netter. So they had their daughter-in-law get breast cancer, and they saw the awful effects of chemotherapy. So they went and said, there must be a better way. And they went to Savio Wu, who said, this is really, cell and gene therapy might be much better, it has promise. And they were wealthy. And Savio put together, similar to ASGCT, he made a CGT, the base pairs in DNA, and it was the Alliance for Cancer Gene Therapy. And that association gave $2 million grants, 2 grants of $1 million each in 2004 to my lab and to Michelle Sadelain. And that's what funded the initial trials that I just mentioned, that it was because neither Michelle Sadelain nor I could get National Cancer Institute to fund those trials because there have been disappointing results for so many years with, cell and gene therapy.
But not really though, right? In the immunotherapy space. I mean, that was, was at that time not quite, I think, recognized, the potential at least, for that. So obviously, you picked HIV as your Trojan horse to get the CAR in. Others picked just the gamma retroviral vectors. Was that because of your HIV background or knowledge or at that time?
It's exactly right. So I'd worked a lot with HIV, and then we found that it caused less silencing. I mean, it evolved, to be expressed in human T cells. And we saw in some experiments we did, silencing with gamma retroviruses. And so that's, why we went with HIV. And we also spent, Mike Malone, who was a postdoc in my lab, spent 2 years just studying different promoters in T cells to maintain, expression of the transgene. And that was a really important issue too, to not have, to have stable expression of the CAR molecule.
Yeah. So maybe I fast forward a little bit now to your first patients, patients that had chronic lymphocytic leukemia. Maybe you can tell us a little bit how that came about, that study, and of course that enormous success then.
Yeah, that was, it came about because of that philanthropic grant from ASGCT. And we used, the protocol was written for CD19 CARs for patients with untreatable CD19 positive tumors. And just by happenstance, our first 3 patients had CLL. Partly is that they could live long enough and have, because the manufacturing took us a long time to get things approved, and so acute leukemia was much harder with a patient and timing, as you know. And so we didn't initially treat ALL. We treated, chemo refractory CLL. And, and the first 2 patients treated in 2010, survived for more than a decade now with no recurrent leukemia. And so just last year, we published a paper showing the 10-year survival of those patients that's leukemia-free, and that the CAR T cells persisted for a decade, which is something we never anticipated because that CAR molecule has a mouse antibody.
Right.
It's a hybridoma called FMC63, and we thought it would be rejected. In fact, in our consent form, we said we thought that the CAR T cells would last about a month based on some results that Stan Riddell had done. And as well know about the immunogenicity of cells in patients. So you spent a lot of time studying that. And we thought they'd be gone. And it turns out by targeting B cells, you could induce tolerance to a mouse protein. It was a really unexpected result.
That was really amazing, those results. Yeah. But also, that these CAR T cells persisted for so long, right? That was, in this particular patient population, because that's not the case for all other diseases necessarily.
No, in fact, so we had in those CLL patients and in ALL, we see the same thing with CD19 targeted CAR cells. They persist a very long time, and in lymphoma. Although part of the reason may be that, they're immunosuppressed to start with, and there's more niches available for lymphocytes in the patients because they've had extensive pretreatment with, B cell and T cell depleting chemotherapy. And we haven't had long-term survival of CAR T cells in solid cancer patients.
Right.
Now, so it's possible that when you target CD19, basically the new B cells that emerge from stem cells are basically acting as a vaccine to boost the CD19 CAR cells. That may be, and so it may be a very special case with CD19 that you have very long-term persistence of the CAR cells. Right. I think where if you actually got sterile elimination of all the CAR targets, with other kinds of, in other tumor settings, then maybe the CAR T cells, unless they're engineered in other ways, may not persist long-term.
Right. So then I think you've treated 3 patients, right? And then Emily came, right? Yeah. She was very sick, obviously. We've heard about this story, and that must have been a very difficult decision, though, at that time.
Yeah, we had, as you said, the CLL patients treated, and then we're able to get with Stephen Grupp at the Children's Hospital in Philadelphia approval to treat refractory pre-B cell ALL in both adults and children. And so the pediatric protocol was led by Stephen Grupp at Children's Hospital, and the adult protocol was led by David Porter and Noel Fry at the University of Pennsylvania. And so, but the first patient, pediatric patient treated was Emily Whitehead in April 2012. And, she had a much more vigorous response than our CLL patients had, probably because, younger age, youthful T cells, and less immunosuppression in ALL than there is in CLL, I think. And so she had very severe cytokine release syndrome. She had 106-degree fever for 3 days and multi-organ failure on ventilator support and comatose. And Michael Kalos, who was in the lab and initially had trained with Phil Greenberg, by the way, he was who did our correlative analysis. And Michael did research labs and found actually that there was extraordinarily high levels of IL-6 in Emily. I mean, basically 1,000 times above baseline. And we couldn't find any evidence of infection. And so my daughter, who has had, juvenile rheumatoid arthritis, I knew about tocilizumab, which had just been approved for pediatric arthritis. And so I suggested to Steve Grupp, why don't you try treating Emily with that? Because she had been treated with very high dose steroids and TNF blockade, and none of that ablated the cytokine storm. But almost immediately after getting IL-6 antagonistic therapy with tocilizumab, she basically effervesced and woke up. It was really astonishing.
Yeah, that was amazing, and that also very lucky that you had that sort of experience with tocilizumab.
Yeah, it was just because of my daughter. I was president of the Clinical Immunology Society in 2009. And as you will do for ASGCT, you have presidential awards. My presidential award at the Clinical Immunology Society in 2009 to 10, which when I was president, was given to a professor in Japan who had, invented the drug. And so he came to the United States for the awards ceremony, and it was really a very interesting. That's the only the reason I knew about it.
It's wonderful, and also to see Emily now, well, she's done. So this was obviously amazing. You've treated quite a number of more patients on this protocol. And then there was the FDA approval. Do you remember where you were and what happened around that time? And it must have been really exciting.
It was. I don't think any of us. You could talk to me or Phil Greenberg or Michelle Sadelain or Malcolm Brenner, who was all, people, that's where they were really working on engineered cells. Yeah. None of us thought it would ever be commercially approved. We all thought it was basically a proof of concept, redirected immune cells and maybe, but after we had those really striking results, Novartis licensed, the CAR T cell we had and began, an international registration trial. And, the results of that confirmed what we had at our single center study. They had in that registration trial, an 82% complete response rate in refractory leukemia. It was really astonishing. And so it was in August of 2017 that the FDA approved it. And I was at the ODAC meeting when I was watching from the audience, and they had, patients who were testifying. Emily Whitehead's father was there, and all saying how it was much preferable to have CAR T cells than chemotherapy and bone marrow transplants. So it was really a courageous thing by the FDA to have approved that, because of many unknowns. It was based on treatment of about 100 patients at that time. And it's probably one of the first therapies to be approved first in pediatrics and then later in adults.
Right. That was surprising to me too. You first treated adult patients. Yeah,
we first treated, but then Novartis. This is another part that's not well known or appreciated is, they have, when you get something approved commercially, you have to be able to show you can treat the affected population ethically, so that there's not auctions and stuff on who gets treated. So they had to be able to treat when they opened up Novartis for commercial sale, the people. And so if it had been a solid tumor, there's no way they could manufacture. The rate limiting step now with autologous T cells is, very difficult in manufacturing. And so with pediatric ALL, there's only several thousand cases a year in the US. Novartis was confident they could do that manufacturing from a single manufacturing center, and then all patients who in the US who had refractory ALL would then be able to get the therapy. So then later, that's why later, as they scaled up manufacturing, they went to adults.
Right. So you say it was surprising. Well, why did you say it was surprising? We knew the immune system can get rid of cancer now from the allogeneic transplantation. We just had to kind of rein in the toxicities and make it more targeted, right?
There's been a lot of surprises. ALL is not, doesn't respond well, as to allogeneic therapy. It's much better with AML, when you use allogeneic, non-gene modified, DLI and T cell infusions and transplants. So it was a surprise that it worked as well as it did. And it actually worked better in humans than it did in mice. Yeah. I think the field's kind of concluded that human T cells are more robust than mouse T cells. So and we never once saw cytokine release syndrome in mice like we did in the humans. There are a lot of surprises, and that's why we need to do phase 1 trials. but the proliferation of the cells in humans, and the other reason I was surprised is, the previous trials had failed. The trials of CAR T cells that were not second generation. So there was one by Cor Lammers targeting carboxy anhydrase 9 in the Netherlands, and then the one that Steve Rosenberg's group did with folate receptor alpha retarget cells. Those CAR T cells barely engrafted and did not proliferate after transfer. And that when we use the second generation CARs with embedded co-stimulation and then improved manufacturing using CD28 co-stimulation, the cells proliferated more than 1,000-fold in all the patients. It was really astonishing compared to basically the lack of proliferation in the previous trials.
So now, obviously, we've got several approved CAR T cell therapies also for multiple myeloma. Now, I'd like to just get a little bit of your thoughts where you think the field is going, of course. Now, we've got CAR NK cells. We've got, of course, CAR macrophages. Where do you think, and you were still trying to figure out things for solid tumors, obviously. Where do you think the field is going with all these different technologies right now that we have available?
I think, and I think you do too, that, cell and gene therapy is a precision therapy that can be ultra personalized. But it's we have the tool set that none of us ever thought, I think. So one major thing, we had the ability to do gene knock-ins basically with lentiviral vectors or gamma retroviruses. But then in 2012 is when CRISPR was first published, and the ability to do multiplex gene knockouts. So that came, we began and did a first trial using CRISPR in patients with three different RNAs and published that in 2020. And now with base editing, so the sky is kind of the limit now in both knocking in genes and knocking them out. So I think there's this confluence of synthetic biology with immunology and better understanding of the mechanisms of resistance to therapies. So I'm very optimistic that, basically all tumors will be treated with some form of cell and gene therapy as we work out the problems. Now, they're not simple. But I do think we're going to see, for instance, also combinations, for instance, various oncolytic viruses used with cell therapies. On clinical studies are very synergistic. So, we now have tool set. The ideas are not new, people were in 1950 in England. There were studies, that by Billingham and Brent and Medawar showing T cells could cause, graft versus host disease. They didn't know how to really grow them, but they could transfer from one mouse to another, and they knew T cells could kill tissues. So the ideas were there at that point then of using T cells to kill cancer cells. But they didn't have the tool set. They just didn't have the tools.
No, it's very exciting. Now we've got the tools, and I agree, And so where do you think, again, this is, of course, right now, it's still a fairly complicated process. I see these patients myself. Sometimes, of course, we have to collect the cells. Sometimes they don't grow. The manufacturing doesn't work, of course. So where do you, if there's allogeneic that people want off the shelf? You've actually done also a beautiful study, with Drew and Jonathan Epstein, Drew Weissman and Jonathan Epstein in vivo, right? Yeah. Where do you think this is going in terms of delivery?
I think we're going to see, so as you just said, there's going to be off-the-shelf cells that are various, allogeneic sources, induced pluripotent stem cells engineered as you've been doing and pioneering. And I think we'll see all of those happen. CAR NK cells, there's about 50 trials now with different kinds of CAR NK cells. And so different cell types, different autologous and allogeneic. Now, the autologous, the logistics are more difficult, but we just reported, at ASH, using 3-day manufacturing, really good results now with an armored CAR that expresses IL-18 and targets CD19. And so when you get down to very short manufacturing, that it doesn't really matter anymore on compared to off the shelf. But what is true is they're much cheaper, to have an off the shelf allogeneic cell line that's used. But the safety is going to be different. I mean, autologous T cells now, Bruce Levine's added up, if you look, about 20,000 patients have been treated worldwide now with CAR T cells, some on, academic trials and others with the commercial products. And at least as of last year, when I looked closely, no cases of transformation were reported with the CAR cells. So there's remarkable resistance of autologous T cells to insertional oncogenesis by gamma retroviruses and lentiviruses. But, in Australia, they reported 2 cases of CAR T cell lymphomas about a year ago in blood. So they had 9 patients treated with allogeneic CAR T cells, and where they had been gene edited, and they used piggyback in that case, a transposase-based system to introduce the CAR. And in 2 of those patients, within several months, they had a lymphoma that had integrated CAR molecules. So they were donor-derived. So that's a very severe toxicity if you have transformation. So, I think one thing for sure is that the allogeneic cells are going to need different safety mechanisms and so on than autologous cells.
What do you think about this study you've done with Jonathan Epstein and Drew Weissman? Could this ever be done? As there's other people interested in that in vivo.
Well, I think that's, the in vivo, there's several approaches, including some investigators at the Fred Hutch on either using in vivo viral directed, where the CARs of cells are engineered in the patient and skipping the ex vivo approach. I mean, in last year, there was a really important seminal paper, I think, where the CAR T cell, virtually identical to the one developed in my lab, was used for 5 patients with refractory lupus in Erlangen. In a study there led by Mackinson, so a long-term friend of mine in Erlangen. And all 5 patients have drug-free remissions. These were refractory lupus patients. And so it worked much better than anyone would have predicted, I think, and what that mechanism is of how restoring self-tolerance by B cell depletion. But so I think this approach of in vivo, what I did with Drew Weissman and John Epstein really is that was targeted lipid nanoparticles to manufacture CAR T cells in mice. And it's quite efficient. I mean, which is really a big issue. Now, the question is, would it be efficient in monkeys or humans when you scale that? And that's, as that's a whole nother issue that we don't know yet. But I think there's going to be in vivo manufacturing of engineered cell therapies. And then long-term, there'll also be this ex vivo use like we're doing now for more sophisticated approaches involving multiple gene multiplexing.
So if I could push you now in 10 years, where do you think we are in 10 years? Now, let's say especially for solid tumors, of course. What do you think immunotherapy will look like in 10 years?
So, right now, basically there's approaches targeting with antibody-based approaches, CAR molecules and then T cell receptors. A huge new part of that is neoantigens, targeting mutated antigens in cancers that have high mutational load. So I think we're going to have both kinds of therapy, FDA-approved CARs and TCR T cells. I think that there's a lot of work ongoing by many labs now looking at how to get around the problem. Because the 2 problems in solid tumors really are lack of good targets like CD19 and BCMA. So that's going to be solved either by combinatorial targeting or switching molecules on and off, like colloidals done on others. I think, but the other issue, is T cell exhaustion that occurs rapidly in solid tumors. I mean, Phil Greenberg studied that at your institute in a lot of detail in mouse pancreatic cancer, and we've studied it in humans and find very similar results that he sees in the mouse. And so the T cell dysfunction and exhaustion has to be overcome too. And I think that that's going to be solved by the field. So I think that may take 10 years. I don't think, I mean, it's a very complex problem of, targeting solid tumors, getting trafficking into the tumors and maintaining the function. And it may require, combinations with cell therapies and viral therapies. But I think the field will solve that. And because now there's so much research, so many labs.
Do you think, we'll need new targets? I think we're both interested in glioblastoma, where obviously that we've thought about CAR T cells, but do we have the right targets?
Yeah, no, and I think, I think what we'll have is, there'll be T cell receptor targets, targeting the mutated antigens. There's hotspots now. Very exciting data this last year with KRAS.
Yeah.
2 patients now have had T cell receptors targeting, the mutant KRAS rather than wild type KRAS, and really exciting results. So I think, we're going to see both the TCR T cells and the CAR T cells, but there'll be, to get, to solve the toxicity, on target, but off tumor toxicity issues, we'll need to either have switchable CAR cells, or, multi-targeting, that are smarts, cells that make decisions on whether to kill or not.
Okay, thanks so much, Carl. So you've just trained so many physicians, scientists, faculty, investigators. What advice do you give to trainees, the people you mentor? What advice do you give them?
Yeah, well, now I think, basically do what you have passion for. And then you just have to follow your nose. There's so many completely unexpected things that have happened in my career, where, and led to other things that were unexpected. And if you follow those opportunities, I think you'll enjoy it. You won't be bored. And the field now we're right at the beginning. I think like in the 1980s, that was just when Microsoft was starting, when I was in Seattle. And then you look at what the computer industry is now. We're like in the 1980s of when in Seattle, when when Steve Jobs was starting Apple and Microsoft. We have right now this same situation in cell and gene therapy. And over the next several decades, it's, I think, going to be an amazing explosion of progress. And so I think it's the most exciting time for young people to go in this kind of science.
That's a great analogy with the computer world. You've also raised 5 children, correct? And how do you balance that family and, your work?
That's impossible. That comes down to each person and their partner coming to an agreement. And, my first wife was a teacher, and I had the same kind of things you did as a postdoc. And I had my first child when I was just getting out of medical school, and she took care, because I was working 100 hours a week. And so that allowed that. And then my first wife passed away from ovarian cancer. I got remarried, 2 more children, and she's a scientist. So we have a much more balanced as far as home, so we've worked that out, with 2 people having jobs, and so that's the hardest thing, I think, is work-life balance. And it's hard in many ways. So, and this career is demanding. Yeah.
Do you think it's gotten harder to do that?
I do. Well, there's been some changes in the medical training that's made that a little bit, more friendly, but there's still a large fallout of women in science because of, it's very hard, with it at least in academia. I think biotechnology industry, it may actually be better for, having family and so on. I think you have better control over your schedule than in academics do. Yeah.
Well, thank you, Carl. So I got to ask you, though, as you know, I'm a biker as well, and I know you're an avid biker, so what do you like to, where do you like to bike these days? Are you still using a gravel bike?
I have both. Yeah. My whole fascination during the COVID thing was because all our races and stuff got stopped. So it began this fascination I got with this extreme event called Everesting, where in one bike ride you climb 29,000 feet, the vertical climb of Mount Everest. And so I did that last August, in August of 21, with my wife and daughter out in Maui. We went up Haleakala. I did 30,000 feet of climbing on Haleakala, and on a bike. And I trained for a year and a half to do that. And I built a special, mountain bike with the right gears to go up a very steep 12% gradient on average.
That is amazing. Yeah, I drove up to the top.
I did it 3 times in 19 hours. Yeah.
Okay. Wow, that's amazing. Do you do any other sports?
Well, I'm running.
Wow. Well, Carl, thank you so much. I really want to also congratulate you on your achievements and really what you've done for patients and really many other scientists. Thank you so much. Really appreciate it.
Thank you. Thanks, Hans-Peter, and thanks for leading ASGCT. It's to see the growth of that organization. I mean, the early days when we were in, it was like every year, could we survive another year?
It's been truly amazing. And of course, I'm very happy to see how the gene therapy, gene editing aspect has really facilitated all these immunotherapies.
Yeah.
Thank you.
Well, thanks again. See ya.